Our first talk is actually from Fabio Vandin, who I think came the furthest distance to be here today. So I'll leave the floor to him.
Okay, thanks. Thanks for the invite. I'm very happy to present some of the work that I've done and some of the challenges that I think there are still to be addressed in finding patterns in cancer genomes. So before starting, actually, somebody made me notice that I had to be at this meeting. So I traveled a long distance. I come from Italy, arrived yesterday night. Just because my name is Fabio and I couldn't miss a meeting that is FAB. Okay? So I also have an hypothesis that that's the only reason I've been invited to this meeting. But hopefully I'm wrong in that. Okay. So we've heard a lot in the first talk from Trace about patterns in cancer genomes and how difficult it is to find them. And so what I'm going to talk about today are a couple of algorithms that we have developed to identify some of the patterns, or at least try to identify some robust patterns in cancer genomes. And to start with, I'm just going to introduce some of the concepts that we need for the rest of the talk. So let's start about talking about alterations in cancer. And so the way in which a tumor develops is we start from a normal cell that is the founder cell for the tumor that during its lifetime accumulates mutations. Some of these mutations are random passenger mutations that do not change the way in which the cell functions and are represented by these black flashes, while some other mutations are represented by these red flashes confer a selective advantage to the cell. And when the cell is hit by this driver mutation, then there is a population of cells that contains all these mutations, and this population of cells a long time will accumulate more mutations. Most of them will be random mutations that not really matter for the disease, while some other mutations confer, again, more advantage to the cell. So now at the end of this process, we have the tumor mass, and what we can do is take it and sequence it. And at the end of the sequencing and of the process that will compare actually the tumor genome with the normal genome, we will measure the somatic mutations in the patient. Okay, of course, when we measure the somatic mutations in the patient, we don't know which mutations are the ones that are important for the disease. We just observe mutation in the entire patient. And now, since the cost of sequencing has been going down dramatically, what we can do is we can sequence a large number of patients and measure mutations or alterations in general for all these patients. And as mentioned by Trey, there are different projects that have taken this approach. For example, the International Cancer Genome Consortium, or ICGC, has released, I guess in April, the last version of the data in which there are alterations released for more than 20,000 samples of different cancer types. So now, using these large data sets and these large sample sizes, we can try to identify the important mutations. But before trying to understand how we can identify the important mutations, these data sets allow us to study the heterogeneity of tumors. And looking at the data, what we can understand is that in a typical tumor, even if they are all different, there are between hundreds and up to even thousands, and sometimes tens of thousands of mutations. Most of them are passenger mutations, so they are not related to the disease. And just a handful of them are actually related to the disease development and have an important role also for the clinic. Okay, so the challenge then that we want to tackle is how can we distinguish the important mutations from the mutations that are not important. And what is done in every large cancer project is to look first for recurrently mutated genes. So we can consider one gene at a time and essentially counting how many samples or how many patients the gene has at least one mutation, compare this number with what to expect if the gene was not important. So compare it with a probability distribution. Usually it's just a binomial distribution. And then if the number of mutations that we observe in the gene is much larger than what we expect, this gene will have a high score in terms of its significance as a driver gene. Okay. So this is a simplified cartoon of what happens in practice, but if you look at large cancer studies, like for example this one from the Cancer Genome Atlas from 2015, you will see a plot that will show the most frequently mutated genes and the threshold for significance. So for example, in this paper, they identified 8 genes that are mutated in a large number of patients. So these are the candidate driver genes, the one that contain driver mutations. And what is not shown in this figure is that there are many more genes, more or less all the other genes in the genome, that are mutated in a small fraction of patients. And for these genes, we don't really know if they are important or not. OK. So I simplified things a little bit in the sense that for this significance score, actually, one takes into account many different features for genes, like, for example, the gene length, the expression level, the replication timing. But all the approaches that have been designed to identify important genes or driver genes by looking at one gene at a time will not be able to assess the significance of the genes that are mutated with a small frequency. So if we look now at the data, we will observe that in many patients, there are no mutations among the one reported as driver mutations in those patients. So in essence, this recurrently mutated genes approach will miss some driver mutations and will not be able to identify all important mutations for cancer. So try then to identify all driver mutations. Instead of looking at the single genes, we can try to look at higher order structures as have been defined by trace. For example, a common approach is to look at pathways. And this is based on the idea that actually the cell is a complex circuitry of interacting genes. And groups of interacting genes perform some function that is important for the disease. So for example, this is just a cartoon. Let's say that we have a group of genes, and these are the genes. So these are the vertices of this network. And we have interactions between genes. And these genes, by their interactions, they perform some function that must be altered in order to derive a tumor, so to obtain a tumor. And there are a number of functions that need to be obtained and altered in a normal cell in order to arrive to a cancer. OK, so now, from simplifying a little bit, any of these genes could be altered in order to obtain the altered function that is needed by the tumor. And so it may be the case that in some large number of patients, only one of these genes is mutated. So for example, is this gene up here. While in the other patients, where this gene is not mutated, one of the other genes is mutated. And these other genes will be mutated with a much smaller frequency than the gene up here. And now, even if the entire set is mutated with relatively or with very high frequency, the single gene approach that look at one gene at a time will identify only this gene as important, and will miss all the other genes that are important as well. So if we want to try to identify all driver mutations or all the mutations that are important for cancer, we shouldn't look at the single genes, but instead we should look for sets of interacting genes, called pathways, that are significantly mutated in cancer. OK, so how can we identify these sets of interacting genes? Well, a common approach in the literature is to first look at known pathways. So there are databases that will define sets of genes that perform some function. And now we can take these sets predefined in some databases and consider each of these pathways individually. For example, we can look at this set of genes, take the corresponding genes in our mutation matrix, in which we have patients on the rows, genes on the columns. And we have one if a gene is mutated in a patient and a 0 otherwise. Now we can count in how many patients this set of genes has at least one mutation. And if this number is large enough, maybe be 80%, then we can use the same statistical test that we have seen before to say that this set of genes is significantly mutated in cancer, therefore is a cancer pathway. OK, so this approach, it can identify some important cancer pathways, but it has some problems. And in particular, not all pathways are known. We don't already know all the possible subset of genes that are related to cancer. Pathways interact, so there is the so-called crosstalk between different pathways, but this approach actually considers pathways in isolation. So they consider them as separated entities. And moreover, some of these pathways that are defined in databases are very large, and only a portion of it, a small portion of it, may be important for cancer. And this approach that look at the entire pathway may miss the important portion if it is not sufficiently large. OK, so what we really want to do is find these pathways, this set of mutated genes, without restricting to known pathways. How can we do that? Well, the first approach that one can think of is to look at possible candidate sets, so subset of genes, up maybe to a reasonable cardinality. So for example, we can restrict to look only at sets of at most 6 genes. And for each of them, we may try to evaluate in how many patients they are mutated and so on and so forth, similarly to what we have described previously. But the problem with this approach is that there are just too many candidate sets. So there are more than 10 to the 20 sets with at most 6 genes. So the approach that I just described would be very computationally expensive, and also it would be suffer from a lack of statistical power in identifying the important sets. Okay, so this is just the setup to start talking about actually algorithms to identify driver pathways, in particular to find driver pathways by looking for specific patterns of mutations across patients of a cancer type. So what we want to do is move from this scenario in which we have a strong prior knowledge regarding what are the important cancer pathways, and using our mutation data, we essentially do a targeted analysis for the pathways that are already known to identify patterns of mutations in the patients and identify, therefore, novel cancer pathways. Okay. Sometimes, in particular for the first algorithm that I'm going to use, we're still going to use some biological knowledge or prior knowledge in the form, in particular, of a protein-protein interaction network. Okay, so in almost all the time that I still have for the talk, I'm going to describe 2 algorithms or 2 classes of algorithms to identify these driver pathways. The first one are the algorithms called HATNet and HATNet2 that find significantly mutated subnetworks of a large PPI network. And the second one is an algorithm called Uncover, is a recent one that finds groups of genes associated with a functional target measure. Okay? So the first approach is essentially an unsupervised approach to identify significant pathways, while the second one instead is a supervised or semi-supervised, it depends on the way you do it, approach to find these important driver pathways. Okay, so let's start with the first one, HATNet and HATNet2. So the problem definition is the following. We're given our binary mutation matrix and the network G that is just a graph in which the vertices are genes and the edges are interactions between genes or the corresponding proteins. And what we want to do is to find connected subnetworks that are mutated in a significant number of patients. Okay? Where significant can correspond to a high number of patients or may correspond to significant from a statistical point of view. So the first thing to notice is that this is a hard computational problem. Okay? So one can actually prove that finding the subnetwork of K genes mutated in the largest number of patients is NP-hard. So even finding just the subnetwork that covers essentially most patients is a computationally difficult problem, but we still want to solve it. Okay, so how can we try to solve this problem? Well, the first approach that we can think of is just to naively enumerate all possible subnetworks of the protein-protein interaction network, and now consider each possible subnetwork as a bag of genes, and now count in how many patients a subnetwork is altered. For example, this set of 4 genes is altered in 1, 2, 3, 4 patients. So if this number is large enough, by using the same statistical test that we've seen before, we could say that this subnetwork is significantly mutated in cancer. Okay. This approach, however, again, has some problems. The first one is that there are just too many subnetworks to test them. So there are more than 10 to the 14 candidate subnetworks that one needs to enumerate and test. And this, again, poses problem from the computational point of view and the statistical point of view, because there will be a lack of power with the number of samples that we have. And the second problem is that the topology of the network is ignored. So we're just looking at the subnetwork as a bag of genes. Okay. So in trying to solve these 2 problems, we have designed the algorithms that we called HATNet and HATNet2. And the algorithms combine the protein-protein interaction data with the mutation data, the alteration data, using a diffusion process on the graph that takes care of properly incorporating the topology of a subnetwork in identifying the significantly mutated network. So just giving you the high-level idea of how the algorithm works, I will be happy to discuss the details if you're interested in them. So the algorithm works as follows. We compute a mutation score for each gene. It could be the frequency of mutation of the gene in different patients, or it may be a different gene score obtained, for example, by single gene tests, if you're interested in those. Then we place on each gene in our protein-protein interaction network a source of it that is proportional to the score that we've just computed. And now we let the heat diffuse on our large protein-protein interaction network. And after letting the heat diffuse, we will have that for each pair of interacting genes, we are going to observe an amount of heat that is exchanged by the two genes. And the amount of heat that is exchanged depends both on the score of the two genes and the topology of the local network that contains the two genes. So after this step, what we're going to do is we're going to remove edges on which not much is going on. That means not a lot of heat is exchanged on that edge. And after removing these edges, we are left with connected components of the new graph that will correspond to our candidate significantly hot subnetwork or the significant subnetwork mutated in a significant number of patients. So arrived at this step, we then have designed a two-stage multiple hypothesis test to rigorously assess the false discovery rate of what we observe. And for the ones that do not know what the false discovery rate is the expected ratios of false discoveries among all the discoveries. So we have a way to bound the expected number of false discoveries in what we are going to report with our algorithm. And this is something that we couldn't do if we were looking at one network at the time, and instead this approach that combines the topology and the score of the network altogether allows us to have a different test for which we can actually bound the false discovery rate. Okay, just to give you an idea of what is that we expect to find by using this algorithm, we used Atney2 on a pan-cancer cohort from the TCGA, in which there were more than 3,000 samples for which we had the alteration data, both somatic mutations and copy number. So we applied our algorithm, and we obtained 16 subnetworks that contain in total more than 140 genes. And what we observe for these subnetworks, so this is just an example, is that some of the genes are really hot. It means they are mutated in a significant number of patients, that they would be picked up even by the single gene approach that look at one gene at a time. But the majority of the genes instead are not identified by single gene tests, and they are actually called as potential driver genes only because we are combining the mutation data with the interaction network by using this diffusion process that I just described. Okay, I'm not going to show you all the results, that show you two examples of subnetworks that we identify on this cohort. The first one is a subnetwork that contains many known genes, for example, TP53, the most famous cancer gene. And so this actually corresponds to identifying automatically knowledge of cancer that is already there and something that was previously known. Another subnetwork that we identify instead corresponds to part of the so-called cohesin complex. That is a complex for which some genes were reported previously to be important for cancer, but its contribution to cancer wasn't as well characterized at the time. So this essentially corresponds to what we thought and we think is a novel discovery for driver mutations in cancer. Okay, so this covered the first part of the talk, and now I'm going to move to the second one, to the method Uncover. So Uncover actually builds on an observation that mutations in a driver pathway usually are mutually exclusive. So if we take a set of interacting genes, as represented, for example, by these 5 genes, and we look at a cohort of cancer patients, and we look at the genes that have mutations in these patients, we observe that for these 5 genes that are captured by the blue boxes here, in most patients, only one of the genes is mutated. And in particular, mutations that are the only one for that patients are in black here. As you can observe, most of the mutations here are black. That means that there is only one mutation among these 5 genes for that patient. While sometimes there is more than one mutations, and they are reported in red in here. Okay? So the observation that mutations in a cancer pathway usually are mutually exclusive is not something that we came up with. Actually, was reported long before we used it. And we deemed it the mutually exclusive property for mutations. And based on this property, we previously designed an efficient algorithm to identify mutually exclusive set of mutations. And we showed that actually they correspond to, in most cases, they correspond to known pathways. So actually, mutual exclusivity is a signal for cancer pathways. And so it's a good pattern to use to identify driver cancer pathways. Okay, after our work, actually, many different methods have been developed that use mutual exclusivity to identify driver pathways, or also to identify the progression at the level of pathways across time for the progression of cancer. And I'm sure I'm missing some of the methods here, but there are many methods that have been developed using mutual exclusivity. But we recently realized that actually, when you look at mutual exclusivity, the assumptions that we're doing is that these mutations are essentially have the same impact at the level of function for the patients. Okay, so the right way to assess and to identify patterns of mutual exclusivity would be to identify patterns of mutual exclusivity that actually correspond to the same effect at the functional level for the cell. So luckily, there is now a lot of data for which we have target profiles that essentially measure some functional, they have some functional measure for different samples or different cell lines commonly. And so we can try to use now these target profiles to do a supervised learning of these mutually exclusive sets, trying to identify then not just sets that show high mutual exclusivity. So for example, this is the set with the highest mutual exclusivity in this cohort of samples. But we can try to identify sets of mutually exclusive alterations that are associated with the target profile. So for example, here we have an association with a high target value. Okay? So this is our goal, to find these mutually exclusive alterations associated with the target profile, either high value or low values. Okay. So let's define the problem a bit more formally. I'm going to go a bit more in detail for this problem, but probably given the amount of time that I have, I'm going to skip some of the details. So the problem definition is following. We have the values for the target profiles. Let's assume that these are sorted by decreasing values. We have a set of samples and a set of genes. For each sample, we have the corresponding value of the target profile, and each gene tells us in which set of samples it is mutated. So what we do using the target values, we also build a penalty for each sample. And then our goal becomes to identify the set of k genes such that the sum of the weight of the elements covered by the genes we select is maximized, so that we have high values of the targets, essentially, and such that the overlap between the selected sets is minimized. Okay. So more formally, given the set of samples, the set A1 up to AN for alterations in gene from G1 to Gn, and the weight Wj for each sample, and the penalties Pj that we derived from the weight, we want to find the set of at most k elements that maximizes this weight, that is just the sum of the weights of the patients in which there will be at least one mutation for the k genes we select, minus the penalty that corresponds to penalizing alterations that are not mutually exclusive. Okay, so not surprisingly, this problem is NPR'd, as most of the problems, or I guess essentially all problems in computational biology turns out to be NPR'd. But we want to solve this problem again anyway, even if it is NPR. So what we came up with is a greedy algorithm to solve the problem. I'm not gonna, the greedy algorithm is fairly easy, but for the sake of time, I'm gonna skip it. I'm gonna tell you just what are the results that we can actually prove for this greedy algorithm. So the first result, we can prove that the greedy algorithm has some guarantees on what it will find. That is, our greedy algorithm will return a 1/k approximation for the best solution. So this is a first encouraging result. However, there is also a result that is not as encouraging. That is that there are instances for which this bound is tight. So our algorithm can be off by a factor k to the optimal solution. Okay? However, what we can prove is that if we have enough samples from a generative model that we think is fairly reasonable, in which there is a set of genes or alterations that is associated with the target values, and the other mutations are essentially placed randomly in the patient, then the greedy will find the set that is associated with the target values with high probability, provided you give it enough samples from this generative model. Okay? Okay, we also designed an integer, an ILP, to solve the problem that works well when the sample sizes are not too large. And we coded this in our approach that we called Uncover, that again takes the target profile, the alterations, and then solve this problem using the optimization problem using either the ILP or the greedy, and performs permutation testing to obtain a p-value for the solution in the reports. Okay, so again, in the interest of time, I'm just going to show you some results on data that we took from the DepMap project, in which there is drug sensitivity data, the target profile for 265 compounds. And there are alterations for 736 cell lines that are our samples. Okay, and so on this data set, we identify around 50 sets of alterations that are associated with the target profile. And these are just two examples. The first one is a set of 3 alterations that are associated with increased sensitivity to this compound, VX11E. And these 3 alterations actually correspond to 3 known cancer genes that are all part of the ERK-MAPK signaling pathway. And so they all make sense that they are associated with the same change at the functional level. And instead, the second result is an association for sunitinib with reduced sensitivity to this compound. And these are 3 genes. RB1 is a non-cancer gene. SETD2 is associated with the genome remodeling. And also, this other gene has some association with a different type of gene remodeling. So this may be some new discoveries on our side. OK, so this was all about past work. And now I just want to tell you what are some of the challenges that I think there are in finding patterns in cancer genomes. And just to make sure, I'm not just talking about the patterns that I described for the work I've done, but more in general for, I think, all the possible type of patterns that people can be looking at. For example, like cancer evolutionary trees that are a really hot area, and I think there will be some talks about reconstructing evolutionary trees for cancer or mutational signature or patient stratification of clustering, as we have heard earlier today. Okay, so the first challenge, I think, is something that I've tried to address in some of the work that I've done, but still there is a lot of work to be done. That is, we need procedure to actually identify statistically significant patterns to make sure that what we observe is not just noise. And the main problem here that, in general, the number of patterns is exponential in the number of features, so we have issues both at the computational side and the statistical side for what is called the multiple hypothesis testing problem. Okay, the second challenge is something that I'm really interested in, and there are some methods that try to deal with it, but not many, regards dealing with uncertainty in the data. And so in particular, the biological measurements that we have and that we use, for example, from sequencing, they have inherent uncertainties. For example, when we call mutations from sequencing data, very often the methods just will use the mutation as 0, 1 values, while instead there are some uncertainties in the process of calling these mutations that are completely forgotten once we try to extract patterns later on. Or even if you do gene expression measurements, usually you try to estimate the expression of a gene, but actually what you see in the data is just a sample from the populations of the cells, for example. And so the measure that you have is not the perfect measure of the gene expression, is some measure correlated with it, and it would make sense to try to use confidence interval, for example, for gene expression. However, there isn't a good framework to try to properly describe and compute these uncertainties for high-dimensional problems like the one we're dealing with. And even less, it's not clear to take into account these uncertainties when we're trying to find patterns. Most of the algorithms, or almost all of the algorithms that I know of for finding patterns in cancer, they really work with data that are assumed to be deterministic. And so the probabilities associated with the fact that the mutation is there or not, or uncertainties and confidence intervals, are forgotten once you try to identify these patterns. Okay, 2 more challenges very quickly. Is one, in trying to identify patterns in complex temporal data. So now you can try to build the evolutionary trees for cancer at different time steps for a patient, and you can try to do it for multiple patients. And now how do you identify robust and meaningful patterns into these data sets? Well, it's not entirely clear. And second, is something that I consider a lot when I do research in my work. It's not just specific to biology, I have to say, but it's the fact that very often algorithm design is driven by NP-hardness. So we try to solve NP-hard problems, and then when we have a problem that is NP-hard, we just look maybe for heuristics. But for biology in particular, sometimes we have an idea of how the data arises. So what would be some reasonable models that is generating the data. And it makes a lot of sense to try to design algorithms that are tied to these models. So I think one challenge for the future is to identify some of these models that are reasonable for different areas of biology and try to design algorithms that are provably good under these models. OK, I guess I'm done with time. I was done 5 minutes ago, probably. So let me just acknowledge the people that work on this project with me, in particular Ben Raphael and Eli Upfal, when they were both at Brown University. Now Ben Raphael is at Princeton. Max Leiserson was a PhD student in Ben Raphael group. Rebecca Sarton, Dorit Ockbaum at Berkeley for the second part about Uncover. And I have a shameless advertising as well about postdoc positions in my group. So thanks. And if you have questions, I would be happy to answer if time permits. Thank you.
Great. So I think we have time for like one question that we can take while Anna is setting up her laptop. Yeah.
So I have 1.5 questions. So one question is, you said you're trying to find subnetworks that have the largest density.
Yeah.
You need to know some bound on the size of the subnetwork, or is that more of a problem?
No, so for the approach that I described, we don't need the bound on the size of the subnetworks. It is determined automatically from the method, from the data.
So if that's the case, then this reduces to something called correlation clustering, which learns the parameter along with it. And there has been some work done where you basically can put positive and negative weights and accommodate all sorts of side information when clustering.
It is not exactly correlation clustering. You can try to frame it as a correlation clustering problem where you say, I know some genes are driver genes, other ones are not, and for some genes I don't really know, and then I can try to find these clusters in this way. One thing is that for correlation, for the problem we have, we don't care too much about all the possible subnetworks that will come out. Most of the subnetworks that will come out are not really important, and so I don't think it can be exactly framed as a correlation clustering problem, but I would be very interested in talking more about that.
The you mentioned are classical parts of the correlation clustering framework. Mutual exclusivity can go into negative weights. Other criteria can go into positive or vice versa. So
yeah, for the mutual exclusivity part, there is somebody that tried to find subnetworks that have high edges with mutual exclusivity. The fact that mutual exclusivity is not really transitive, so it's not clear if you find, for example, a triangle makes sense. But we'll be happy to talk more about it.